We're still continuing with Störinger processes in cells this week. This time we're going to be looking at cell reproduction, the R in Störinger. Let's start with a review of the reasons that cells divide. The first reason that cells divide, you'll need to write this on your graphic organizer, is to maintain an appropriate surface area to volume ratio. Cells need more surface area than volume so that things can get in and out of them efficiently. So a small cell has more surface area to its volume. Therefore, it's more efficient at transferring materials into the cell, things like oxygen and nutrients, and getting waste products out of the cell than a large cell will be. So this surface area to volume ratio limits the size that a cell can become. The second reason that cells need to divide, write this on your graphic organizer, is for growth. As organisms get larger, as now you are larger than you were when you were much younger, you have added cells to your body. Your cells themselves have not gotten bigger, you've just gotten more of them. The third reason, write this in, is to repair or replace damaged cells. If you have gotten hurt, like a broken bone, that bone needs to repair itself by regenerating cells. If you have lost blood, those blood cells need to replace themselves. If you have scraped your knee, the skin cells need to replace themselves. This process that we are discussing today occurs in the body cells or somatic cells of an organism. Write that in the space provided. Somatic is a term that refers to the cells that make up the body of an organism, their skin, their bone, their muscle, their nerves. All of these are examples of somatic cells. You've learned that there are two different categories of cells. Prokaryotic cells are those that do not have a nucleus. And in these types of cells, cell division is very simple. Look at the picture. You have the same picture on your graphic organizer. You start with one simple prokaryotic cell. You can see that there is a chromosome inside. That chromosome replicates itself. Look at the picture and see that. The cell then pinches into two cells. It's a very simple process that occurs in prokaryotic cells. So on your graphic organizer, in the space for prokaryotic cells, write that it is simple because there is no nucleus. The cell simply splits. On the other hand, in eukaryotic cells, it is much more complex. You will need to write in all the bullet-pointed information shown on this slide. Eukaryotic cells are more complex because they have a nucleus. This nucleus contains chromosomes. There's only one nucleus in a eukaryotic cell. So in order for that nucleus to be divided, the chromosomes first have to make a copy of themselves. They have to be duplicated so that the new cell can receive a copy of them. This makes the process of cell division in eukaryotic cells a lot more complicated than the process of cell division in prokaryotic cells. So the special name for the process in eukaryotic cells is mitosis. You have a place to define mitosis on your graphic organizer. The process of nuclear division in eukaryotic cells. Nuclear referring to the nucleus of those cells. Next, we're going to look at the life cycle of a eukaryotic cell. Remember, we are specifically focusing in on the division of eukaryotic cells because it's so much more complex. The cell's life takes place in phases. The longest phase is called interphase. Interphase, you have a space to write this in on your graphic organizer, is the normal life of the cell. During this stage, the cell is growing. This occurs in phases G1 or growth 1 and G2, or growth 2, of interphase. Notice on the circular chart beneath the word cell cycle that G1, S, and G2 are bracketed, meaning that all of these are part of the phase called interphase, the normal life of the cell. During the S phase of interphase, the DNA is copied, and that is hugely important. Make sure that you've written all of this bullet-pointed information in the space provided for interphase. Now let's take a quick look at how this DNA is copied. Look at the picture of the chromosome that is shown on this slide. You'll notice it looks like an X. Each half of the X is one copy of the DNA. Each half of the X is called a chromatid. Please make sure you are drawing a doubled chromosome in the space provided, labeling the two chromatids and the centromere, which is attaching the chromatids to each other. Again, it's very important that you recognize that this doubled chromosome is a doubled copy of the DNA. As the cell is preparing to divide, it has to make a copy of the DNA so that it can give that copy to the new cell being produced. Now let's move to the other side of the circle where you see mitosis bracketed by itself. You have a space to write some information about mitosis. Mitosis is the process of eukaryotic cell division, which makes an identical copy or clone of the parent cell. So at the beginning of the process, you have a parent cell with a particular number of chromosomes. In this example, that number is 46. That's representative of humans. Humans have 46 chromosomes in every cell of their body. After the process of mitosis, you end up with two daughter cells, each of them containing 46 chromosomes. This is possible because before the process started, the DNA was doubled. Make sure you have written in the correct information on your graphic organizer. Now look down beneath the box labeled mitosis to the pictures of the process. You need to label the name of each picture. These are the phases of mitosis. Mitosis is the actual division of the nucleus. So it starts with prophase. You'll see that the chromosomes are not doubled. They don't look like X's yet. In prometaphase, the chromosomes have begun to look like X's because the DNA has been copied. In metaphase, those chromosomes line up in the middle or at the equator of the cell. In anaphase, those chromosomes are being pulled apart. The X's are being pulled apart. One side of the X goes to one side of the cell, the other side of the X goes to the other side of the cell. Remember, each side of the X is called a chromatid. So half of the DNA is moving to one side, half of the DNA is moving to the other side. Lastly, we have telophase, where the cell itself actually pinches and you end up with two identical daughter cells. Now you need to write in at the bottom in some blank spot that you have available that there is a final division that splits up all of the material in the cytoplasm. It's called cytokinesis. That is not pictured here. You need to add it into your diagram, maybe underneath it. Cytokinesis is the final division of the cell. Sometimes mitosis goes wrong. The DNA in the cell is what tells the cell when to divide. Sometimes there's a problem with that DNA. Maybe there has been some sort of a mutation, and the signal that tells the cell to divide gets messed up. So it divides uncontrollably, leading to a growth of a tumor or cancer. Not all tumors are cancerous, but some are. This is a result of uncontrolled cell division, leading to cancer. Now remember, we said that cells need to divide so that they can stay small in size, so that organisms can repair or replace lost or damaged cells. Cells need to divide so that they can allow an organism to grow. But there is another reason that cells need to divide, and that is for reproduction of an entire organism. There's two kinds of reproduction. The first is asexual. Look on the back of your graphic organizer. Fill in the blank at the top. Reproduction of organisms depends on cell division. Starting by looking at the asexual form of reproduction. In this type of reproduction, there is only one parent. Write this in. The offspring, therefore, are going to be identical or clones of that parent. So look at the diagram. If the parent organism contains 12 chromosomes in the cells, the two daughter organisms will have 12 chromosomes in each cell. It does not split. 12 and 12. The process of cell division that allows this to happen is mitosis. Write that in. So all of these types of asexual reproduction that we're about to go over are possible because of mitosis. Now in your notes you were asked to draw some simple pictures. You probably had a difficult time doing that, so you should open those notes back up now so that you can draw these simple pictures in the spaces provided. You also need to list these types of asexual reproduction on your graphic organizer in the space for asexual reproduction under 1 through 5. The first one is binary fission. Binary fission occurs in single-celled or unicellular organisms. This occurs when cells simply split in half. The picture is shown for you. The next type of asexual reproduction is called budding. Budding also happens in very simple organisms like the one pictured, which is called a hydra. A hydra is a microscopic organism, but it is made of more than one cell. You can see that the hydra, adult, has a tiny little baby budding off of it. That bud is going to grow into an adult hydra and separate from it. It is a clone of the parent. The next type of asexual reproduction is sporulation. Sporulation occurs in fungi. The production of spores is what's being referred to here. The picture you see is bread mold, and the stuff up at the top popping out of the little round stalk is the spore. There are several spores actually. They look like dust. If you've ever kicked a mushroom or run over a mushroom with your lawnmower and noticed that there was a bunch of dust that resulted, that dust is actually spores. And those spores will grow into new baby fungi. Sporulation. The next type of asexual reproduction is vegetative propagation. Vegetative propagation occurs in plants, hence the term vegetative, which refers to vegetables or plants. Vegetative propagation occurs when plants produce clones of themselves. The plant pictured here is a spider plant. The spider plant sends out runners or stems with little babies at the end. These little babies are clones of the parent. They will grow into new plants. The last type of asexual reproduction is regeneration. Regeneration occurs when an organism is able to regrow lost parts. The picture shown is a starfish, which has lost a leg, and the leg is regrowing. There are also worms that, when cut in half, can grow into new worms. Regeneration, again, is possible because of cell division called mitosis. There is another form of reproduction. This type of reproduction is sexual. So looking at your graphic organizer, there's a place to write in information about sexual reproduction. In sexual reproduction, there are two parents, meaning that the DNA of the mom and the DNA of the dad is going to get mixed up. Therefore, the offspring will be different from either parent. This requires a different type of cell division. This type of cell division is called meiosis. Meiosis is the type of cell division that has one job only. The job of meiosis, and there is a place to write this in, is to produce gametes. Gametes are sperm and egg. Sperm is the male gamete or sex cell. Egg is the female gamete or sex cell. So notice that the process of meiosis is different than the process of mitosis. If you start with an organism that has 46 chromosomes, like a human, there are actually 2 divisions to produce these gametes. In the first division, that number is split in half, 23 and 23. Therefore, it's called reduction division because it reduces the number of chromosomes by half. Then, each of those cells is split again. So you end up with 4 gametes, each with 23 chromosomes. It does not split in half a second time. Make sure you filled in the numbers correctly on your graphic organizer, showing that you start with 1 cell with the normal number of chromosomes, and you end with 4 cells with half the normal number of chromosomes. So let's look at why we need the process of meiosis or meiosis in a little bit more detail. This can be complicated, so please focus. This is also on your graphic organizer in the sexual reproduction section under meiosis. The reason we need this process of meiosis is because of fertilization. Fertilization is simply the process of egg and sperm coming together. This is pictured here for you. When a sperm cell connects with an egg cell, that is called fertilization, and the DNA of those 2 cells is mixed up. So we start with the egg cell. It needs to have half of the normal number of chromosomes, as does the sperm cell. It needs to have half of the normal number of chromosomes, because when they come together, you get the whole correct normal number of chromosomes in the zygote. So you should write the number 23 in the egg cell, the number 23 in the sperm cell, and the number 46 in the zygote. Underneath those pictures, you need to write the words egg, sperm, and zygote. The words haploid and diploid are already written in for you, but please define those words. A haploid cell has half the normal number of chromosomes. A diploid cell has the normal number of chromosomes, 2 of each kind. The number of chromosomes is different for every living organism. Humans have a normal number of chromosomes of 46. Other organisms have different numbers of chromosomes in their cells. So let's look at the pros and cons of meiosis. Meiosis, and there is a place to write this in, is not as fast as asexual reproduction. This means that organisms can't produce as many offspring. They need to find a mate in order to have offspring. But the benefit to this process, the pros of this process, are the genetic variation that is produced. Because the DNA of the mother and the father are being mixed together, this means that there is genetic variation. Clones are not being produced. We have totally different new organisms with genetic variation. Let's look at this genetic variation that's produced in meiosis. You'll see that coming down from the pros and cons section of your graphic organizer is a big fat arrow. Please write the words genetic recombination in that big fat arrow. Then look over, starting at the left side of the diagram. You will see that starting on the left side of the diagram, some of the chromosomes are light colored and some of the chromosomes are dark colored. You will also see that during the process of genetic recombination, pieces of those chromosomes have switched places, so that there is a dark piece on a light chromosome and a light piece on a dark chromosome. This is called crossing over. There is a line to write that term. Crossing over occurs, define it, when homologous chromosomes switch pieces. This is an example of genetic recombination. It is one reason that you get genetic variation when the process of meiosis is used to allow for sexual reproduction. Another way that you get genetic variation in meiosis is through the process of independent assortment. And you have a space to write this in as well. Go ahead and define it first. Homologous pairs of chromosomes are sorted differently each time a gamete is formed. Humans have 46 chromosomes, but they occur in homologous pairs. The prefix homo means same, so there are 23 pairs, 2 of each kind, and they line up in a different way every time the process of meiosis is occurring, so each of the gametes is a little different from each other. Sperm from the same man and eggs from the same woman can have different combinations of genes because of this independent assortment. So let's look at an example of a parent cell. You have this parent cell drawn already for you on your graphic organizer. You need to go ahead and draw in 4 small circles beside it. In those circles, we're going to show how these chromosomes can get sorted differently. In the first small circle, you need to write big S, big T. That's one combination that could happen. Another combination for you to write in the second circle is big S, little t. So that's different. In the third circle, you would write little s, big T. And in the fourth circle, little s, little t. The S's and the T's represent genes on those chromosomes. A big S and a little s represents a different form of the same gene. A big T and a little t represents a different form of the same gene. You can see how from one parent cell, there are 4 possible combinations of these genes. That is another reason that during the process of sexual reproduction, which occurs because of meiosis, we can get genetic variation.